Our next presenter is Dr. Ben Atkinson, representing Flagler College.
Thank you for the introduction. I want to start by saying that this is primarily an image-driven narrative that will highlight our questions, methods, and some preliminary results. So I've got a lot of partners, and I want to say first that this is a true collaboration with my undergraduate mentees. Many of you may be aware Flagler College is an undergraduate institution, and I think it's part of what makes us special, getting undergraduates involved in research. You'll get to see photos of many of them throughout the presentation. And at the end, I'll name several of them specifically if I haven't already. But before that, I just want to mention Flagler, the Florida Undergraduate Research Association, GTM Research Reserve. We've had some funding from the Women of Vision Fund. Obviously, we're permitted through FWC. And I wanted to highlight a colleague, Mr. George Heinrich, director of the Florida Turtle Conservation Trust, has been a friend and colleague and really helps guide a lot of the directions my research goes. And then Dr. Anthony Lau, which you'll hear about a little bit more later toward the end, went to graduate school together, and he conducted work on the tortoises in these dunes long before I did. And then finally, the Gopher Tortoise Council. So there's been some related studies. First, we're lucky to have some nice books on gopher tortoises to go back to, a sort of overview of literature by Ray Ashton, Patricia Ashton. Biology and Conservation of North American Tortoises is a great resource. And then even more surprisingly, perhaps, given how little has been published on gopher tortoises in these coastal uplands and beach dune habitats, we actually have no less than 3 papers specifically on the habitat site that you'll see a lot about, our North Beach dune sites. So Pollock and Kimball, Lau and Dodd, and Blonder et al. Are all papers that have preceded our work. So most of you are familiar with gopher tortoises and know that they live in burrows. You can see on the left, I'm pointing toward a burrow at the North Beach dune site. We can see a tortoise inhabiting a burrow on the right. And these burrows are really important. They've been documented to be a source of refuge for over 365 other species in other habitats. So I should say with a caveat that most of those research studies, most of those data have emerged from longleaf pine studies and other similar habitats. The truth be told, we know very, very little about those commensal organisms and those other species that will go in and out of their burrows in these coastal habitats. So this is a nice little diagram from the Gopher Tortoise Council, and we can see some of these on the top left, casual visitants. We see a rattlesnake, a skunk, a possum. And then we see some true commensals, so species that are really dependent upon those burrows, with the race runner, the gopher mouse, the gopher frog, cave crickets. And then we're going to highlight a little bit later in the slides today a relationship between these ticks and tortoises. And so if anybody attended the poster session last night, you may have seen a poster by some of my students on ticks and tortoises. You may have seen a poster that also focused on using a burrow cam in a robot to look down inside them, and I'll talk more about that in a moment. So most of you are familiar with where the GTM NERR is, or the GTM Research Reserve, and you can see on the left a map from Lau and Dodd, 2015. It shows the general gist of where it is. We can see a more detailed map in the center that specifies these different parcels. And then on the right, you can see with the yellow push pins, our dune site at the North Beach Guana River Preserve in the north, and then our peninsula site down behind the GTM Visitor Center near the trailhead. So this is a kind of an aerial view of the North Beach dune site. It's a coastal dune habitat. The sediment is fine quartz sand. And if you look, each of these blue teardrops with numbers represents a tortoise that we tagged last semester in that habitat. It is bounded on the west side by salt marsh and a road, and on the east side by the ocean. So these tortoises are kind of squeezed in the middle of that, but apparently thriving in this dune habitat nonetheless. Most of you have probably, if you've been to the GTM a time or two, have actually walked along Guana River Road. You may recognize the weir in the forest beyond. So this is behind the GTM Visitor Center. We refer to it as our peninsula site, and it's got some upland forest. The sediment is shell, hash, and clay. And the reason that I mention these sediments is it plays a particularly important role in the relationship that we're documenting between the tortoises and their ectoparasites, or the gopher tortoise tick. So we do a lot of the sort of standard turtle nerd stuff, mark recapture insights here. We take measurements using tree calipers. You can see Shane Smith here with aluminum tree calipers, Aaron Beal holding the tortoise, and Cassie Picard taking photos on the left. All students that were enrolled in my capstone course last semester that focused on tortoise research. These shell dimensions are sort of standard issue stuff. If you study turtles or tortoises, we take the carapace length, or the top shell length from front to back. We do the carapace width, the plastron length, which is the belly shell length, and then the height of the turtle. And you can see Shane's preparing to measure the height of this animal on the left. We can also take the mass. So you can see this little juvenile tortoise on the right. We're collecting mass using a digital scale. Obviously, we need a bigger scale when we measure bigger tortoises, but these are pretty standard issue things. The other thing we do is pit tag the animals, and that stands for passive integrated transponders. And we use a pit tag reader to read their numbers. If you look here, you can see a little yellow reader on the bottom left, on the left-hand photo. We also count growth annuli, so we try to get age estimates on each of these animals in addition to their size. And we've measured and tagged animals from as small as the little one you saw on the scale in the preceding slide to this big mama on the right, which happens to live near the bathroom by the trailhead. If you've been down there, you know her burrows roped off with survey tape. And this allows us, by tagging these animals individually, to track their growth over time, to identify them again if we find them, and it also helps with some of our parasitic research as well. So I mentioned parasites. These are gopher tortoise ticks. You probably might get the heebie-jeebies when you see these things. Most people start to instinctively scratch their arms and legs whenever they see ticks, me included. But my student Shane Smith noticed that our tortoises in the dunes had a lot of ticks on them. And over time, we started to notice that we didn't really see ticks on the ones at the peninsula site. And so this whole area of inquiry, I have to attribute to Shane's, being Shane's brainchild, not mine. But I'm happy to go along for the ride, and it's been really interesting stuff. So we've been identifying those ticks. We pull all of the ticks off of a given tortoise. They get preserved in alcohol with the pit tag number for that individual, so we know which ticks belong to which tortoise. If we catch that tortoise again in the future, we can go back and say how long did it take, perhaps, for ticks to kind of recolonize that animal. So here's some preliminary data we can see on the left. The tortoises in the dunes having a relatively high tick count. So one individual in particular had 15. And on the right, we see no ticks in the peninsula site, which came as a bit of a surprise. The other thing that we're seeing is that the number of ticks is very strongly correlated with the size of the tortoise, as you might expect. So the littlest tortoises may have no or few ticks, but as they get bigger, they collect more parasites. So we can even break that down. This is maybe perhaps a little counterintuitive. We're starting from big and going to small, left to right. But we see adult tortoises on the left, subadult in the middle, and juvenile on the right. If you look at the colored dots, you'll notice that there are reddish, pinkish dots for the dunes and blue dots for the peninsula. All of the blue dots, you may notice, are down on that 0 line at the bottom to represent the fact that we didn't have any ticks on any of the tortoises from the peninsula site. But you can also see that correlation where the bigger the tortoise, the more ticks they had on their bodies. Another fun area of inquiry that we've gotten involved with recently has been using a robot and a burrow camera. And you can see Abby Belshore on the right with a remote control and this little robot at the dune site, and then on the right there at the peninsula site. And basically, this robot is conveying our burrow camera, which is this normal burrow camera, but it keeps it upright It allows it to not disturb the sediment so much. And it gives us much clearer images. And then our Burrow camera now is also paired with a digital, a DVR, so that we can record images and get much higher quality stuff for analysis later. We're interested in knowing what kind of commensals live in these habitats. No one's really ever explored that question. What other critters are using these burrows in this habitat, which is very different, obviously, from a longleaf pine forest. We've also set up trail cameras, and this has elucidated some pretty interesting stuff. If you look on the left, you see a gopher tortoise on its apron. We've been fortunate to get really neat stuff about activity patterns, looking at temperature. These cameras actually collect temperature, so it lets us know a better idea of when they're active and helps us maybe better plan our sampling efforts. If you look very carefully on the right, you'll even see a coach whip right here coming out of the burrow, and I've got multiple images now of that coach whip coming in and out of that burrow. It seems to spend a lot of time there. We've gotten a lot of mammals that come around. You saw those on those casual visitants at the slide at the beginning, with deer and squirrels and armadillos and raccoons paying visits, but we haven't documented them going into the burrow. At night, we see sometimes a different cast of characters. It was a little unusual to see that raccoon in the daytime on the preceding slide. We see a lot of them at night. You can see this is a curious raccoon on the left. And then on the right, the whole thing that really started this trail cam investigation was we saw quite a bit of depredation of tortoises occurring in the dunes. We've collected skeletal material, I'll show you in a moment. And we saw bobcat tracks and coyote tracks and scat. And at one point, you can see if you look carefully on the right, a bobcat that we finally captured in the dunes. So that led us to start to salvage skeletal material that we found. We wanted to know a bit more about that. And I had a background with skeletal material and allometry. We took some papers from Dick Franz and Irvi Quitmeyer, and were able to conduct allometry based on even on incomplete skeletal material. So you can just have, in some cases, a reference bone, a piece of the plastron, for instance, and extrapolate what the body size would have been based even on a single bone. And so that tells us a lot more about what we're missing.
3 minutes.
Thank you. So we can look at the living tortoises in the dunes, and then and the living tortoises in the peninsula on the right. We see frequency on the left and the size classes along the x-axis in millimeters. That's the straight carapace length is a common indicator of body size. But what you might find interesting is if I advance this slide, all of the things featured in red here are animals that are represented by the skeletal material. So you've got the preceding slide that you saw like this with all the living ones, but now we can add in what we're missing, which demographic categories have been recently depredated. And we see quite a lot of depredation occurring again in those dunes. It appears to be related to perhaps coyotes and maybe even bobcats. We'll hope to find out more about that in the future. You also notice reproductive adults missing occasionally as well. So those were the biggest shells in the slide I showed previously. So you might ask, what's next? If you look carefully at these shells, we see some old epoxy here and some drill holes. These were actually tortoises that were captured and marked and radio tracked by Dr. Anthony Lau, who I mentioned earlier in the slideshow. We went to graduate school together, and he kindly provided all of his raw data from graduate school, which now unlocks a lot more information for us. So if we catch an animal, we can go back and figure out how much it's grown since then, how much it's moved, maybe get a better estimate of how old it is. And I find that absolutely fascinating. If you study long-lived animals like turtles and tortoises, you need to work on them for a long time to get true insight. And so long-term ecological monitoring is important for lots of questions.
1 minute.
But it's particularly important when you study something like turtles and tortoises. And so I just want to mention again that I've worked with a lot of undergraduates. They're very enthusiastic. We've sort of dubbed ourselves the Tortuga crew. So you can see them holding the flag in the middle. But I especially wanted to mention Cassie Picard, Abby Belshore, Shane Smith, Avery Cogley, and Aaron Buell, who have worked the most of my students on this tortoise research recently. And so with that, I believe I am out of time, and I will take any questions that you might have. And I can see quite a few in the chat bar here.
Yes, thank you, Dr. Atkinson, for that great presentation and a great overview of all the research that's going on with you and your undergraduates. We had a few reactions to the photos of the ticks. And there was one question: do you know how much time the tortoises spend inside versus outside their burrows?
So that's entirely dependent on the time of year and the time of day, and it's really a temperature related answer. So like almost anything in ecology, the answer is it depends. It depends on time of year, depends on the temperature. We are finding that they tend to be a lot more active when it hits at least 75 degrees Fahrenheit, 80 degrees Fahrenheit, even more so. But the depth of the burrow can affect that. It's when they're way down in the bottom. It's a matter of that place warming up enough to draw them out sometimes.
Definitely. With the difference in the ticks between the dunes and the peninsula, do you know if there may have been maybe more potential hosts on the peninsula that the ticks would prefer other than the tortoises? Do you think there's a difference there between the peninsula and the dunes?
Well, certainly, if our trail cams are any indication and tracks and stuff, there was an awful lot more biodiversity that's frequenting the burrow areas in the peninsula site. It's lots of mammals and very little activity, frankly. We had that camera up for 3 weeks or something, and we got one bobcat and one raccoon at the dunes. But it's also this gopher tortoise tick for certain stages of its life. The tortoise is its real primary host. So I think we've got a lot to learn yet. One of the main things that we're going to transition toward is looking at soil, so soil type and sand depth. There was a paper by Ennin and Qualls that actually did a really nice job in Mississippi showing areas with and without gopher tortoise ticks and characterized the soils. And so we need to replicate that here. And I think, frankly, that's probably going to be the most, potentially the most promising line of inquiry to help us understand this association.
Great, thank you. There were a few questions about the ticks specifically. Do you know if the ticks carry any diseases that are harmful to other animals, the tortoises themselves, or humans?
That's certainly outside of my area of expertise, but we're exploring the tick literature. Surprisingly, there's very, very little specifically on the gopher tortoise tick. So on this particular tick that we're finding of interest, there's not a lot known. With other species, things like Lyme disease and Rocky Mountain spotted fever and all that kind of stuff are things that concern. Certainly, we check ourselves well when we get down in the field and try to make sure that we don't put ourselves at unnecessary risk.
Did you see any of the commensal species or the tortoises react to the burrow bots when you were doing that research?
Yes. First of all, I think they seem a little surprised whenever something shows up inside their burrow that looks foreign, and particularly whenever it has little lights. So there's little LED lights on the front end of the camera, and all of a sudden there's light down in an area that is typically always dark. I think that surprises them a bit. A lot of times, if you're not using the burrow bot, they just turn around. You bump into it before you see it. And so you end up just seeing a tortoise butt, for lack of a better way to put it. But what we have noticed with the burrow bot being quite a bit less invasive and easier to maneuver, we just had one have a staring contest with the burrow bot the other day. So it just looked at it. It didn't seem entirely phased, but it was certainly curious.
Awesome. There are a few more questions that we will not have time for today, but one quick question. Do you know how old your oldest tortoise is that you track?
That's a fun question. I would feel like a little premature answering that, but given now that we have those data from Dr. Lau, and that we can say, how old they estimated to be at that time and then add another 10, 12 years, I think we'll have, hopefully for next year's State of the Reserve, I'll be able to tell you more about that.
Awesome. Thank you so much, Dr. Atkinson.